Hi, my name is Grace Greenway, and today I will be discussing Ras. Ras is a protein that signals for cell growth in eukaryotes. And we're interested in Ras because it is hyperactively mutated in 33% of cancers. A little bit of background on Ras. It is in the CAAX family of proteins. CAAX refers to the final four amino acids of the protein, which are cysteine followed by two aliphatic amino acids and then a final amino acid that can be one of many. These proteins undergo a series of post-translational modifications, and I'll go over those very briefly, but they are outlined right here. First, the cysteine is isoprenylated, then the final three amino acids are proteolytically cleaved, and finally, the cysteine is carboxymethylated. Now, this is an addition for Ras, but it is a CAAX protein specifically, which means there's a cysteine that precedes the CAAX box. This cysteine is palmitoylated in addition to the rest of the modification pathway. These modifications influence the localization of the protein. It usually resides at the plasma membrane. In this study, we propose that interruption of the CAAX-PTM pathway can alter its localization. This study focused on how the CAAX-PTM pathway affects Ras signaling and localization in a yeast model. The purpose of our investigation was to target the proteolysis step. We hypothesized that if this step is disrupted, signaling and localization will be impacted. We investigated function through a heat shock sensitivity assay. The premise of this assay is that when hyperactively mutated Ras is localized at the plasma membrane, the cell dies under heat shock. In this assay, we investigated how the proteolyzed status of Ras affected its function. Our experimental strains consisted of strains where proteolytic enzymes were deleted, such as the one circled here, as well as a strain that harbored uncleavable Ras. From these results, we see that strains that remain unproteolyzed survived the heat shock. And an important point to make is that the strain where only Ras was unproteolyzed performed most like the positive unmutated control. Next, we investigated localization through GFP tag microscopy. In this study, we compared three variants of Ras, each of which underwent the PTMs to a different extent. Additionally, the wild type of each variant was compared to the hyperactive mutant of that same variant. This mutation in yeast is a mimic of the mutation seen in some Ras-related cancers. We'll begin with the variant seen under A. This variant undergoes the entire PTM pathway. The wild type of this variant is localized at the plasma membrane. However, the mutated version has very low Ras expression. Our next variant is only isoprenylated. It does not undergo proteolysis or carboxymethylation. In both the wild type and the mutated version, the protein is localized in the cytoplasm. Our final variant, under C, completes the regular CAAX-PTM pathway. However, it is not palmitoylated. In both the wild type and mutated versions, the protein is localized on internal membranes. From these results, we can make a few conclusions. Based on the heat shock results, we see that yeast survivability increases when Ras2 G19V is not proteolyzed. The effect of Ras2 G19V therefore decreases when proteolysis is lost. From the microscopy results, we see that loss of proteolysis mislocalizes Ras2 G19V from the plasma membrane to the cytoplasm. We hypothesize that this results in decreased interactions with upstream activators and downstream effectors. Now, why is this important? Well, one thing to mention is that an important step to developing cancer therapy is understanding the mechanism of the cause. In understanding how the modifications of Ras affect where it's localized and how that affects its signaling ability, it brings us one step closer to understanding how this disease works. Now, the relevance of this research specifically is that the cleavage step of the CAAX-PTM pathway could potentially be a therapeutic target for human cancers involving Ras. Thank you very much.